All right, let's get going. So this lecture is 3.3, focusing on the different classes of T cells. So we're going to go into all the different types. We have a pretty wide variety, especially when compared to B cells. A lot more distinct behaviors to discuss here. But let's just talk about it broadly. Okay, so the T cell role, they're field generals of the adaptive immune system. They fight infection for us and coordinate and regulate nearly every immune response. So unlike B cells, your T cells are out there, the CD8 especially, with that cell-to-cell contact, getting rid of any intracellular infections. And the CD4 cells more so with the signaling and orchestrating of everything. So what are they important for? Well, obviously intracellular threats, I already mentioned that. So viruses, some bacteria, like we talked about in the beginning of the year, opportunistic pathogens like E. coli are really adept at forming intracellular bacterial communities. It's really difficult for other immune cells to understand that your own machinery has been overrun, but these cells are capable of detecting when something's not quite right inside. Also, tumors are a great example of the benefits from these cells because, well, tumors, again, they are made up of our own molecules, our own self-identifiers. It's still our MHC, more or less, but whenever we have infections from a virus or overrun by a bacteria, or we have unregulated replication and differentiation, well, what that's going to do is it's going to mess with the MHC a little bit, or it'll mess with some sort of surface detection that these T cells detect as wrong. So they are excellent at bypassing what could just look like a regular old one of your cells, but they are able to understand that something is going wrong internally. And again, a lot of that's due to MHC, and we have a whole day on that next video. Also great for coordination. Again, more of the CD4 side of things, signaling the B cells on what to do. Maybe what antibody I need. Maybe I need IgA. Maybe I need mucosal defense. Maybe I know it's an extracellular bacteria. I need a lot of opsonization. Or maybe for some reason I got a dang worm. I need a multicellular parasite. All right, so that could maybe be a function there, detecting what I'm fighting due to what signals that are in the body, and then I can have my T cell tell my B cells what type of antibody I need. And great for regulating inflammation, so that's going to be more so on the T regulatory cell kind of things. All right, so already 3 distinct classes that I'm talking about here, and that's not even all of them, unfortunately. So their action is tightly controlled. Remember how many T cells don't make it past selection checkpoint. 95%. And I think that's honestly a generous number. I think I've seen somewhere around the percentage of only like 2 or 3% make it through. So 95% do not make it through T cell college. Crazy, crazy stuff. But that's important because if those T cells are insufficient, well, then we have chronic infection. Then I can't beat any virus. I literally can't beat any virus. Or if I'm overreactive, bang, autoimmunity. I am, wherever the T cell that is autoreactive has taken residence, then that organ is going to have a mighty tough time. All right. Or maybe it's a CD4 T cell, and maybe it has some sort of dysregulation in its secretion pathway, and now I'm getting all of these cytokines, all of these signaling factors that I didn't ask for. And if you recall, right, I don't want to go too heavy on cytokines in this class, but this is how these cells communicate. They don't have ears, they don't have eyes, they don't have different dialects. Their dialects, their speech patterns are these cytokines. So if they're overrun with this cacophony of different languages and different volumes of speech, well, then I have complete disarray. I have no idea, my cells have no idea where to go, no idea what to fight, no idea where to defend. So it's a whole mess. So that's why we're so selective. 95% plus don't need to be out in circulation. I need the best of the best. So these are my cards, just to include here our main class of T cells. We have our CD4 T cells. I like to represent that with the orchestrator, sending all those molecules, those signaling factors out. Our CD8 T cell, I have him represented as an assassin with his daggers going straight into cell-to-cell combat, digging those viruses out of our own cells. And then our T regulatory cells represented by the scales of balance, keeping everything in check, because sometimes we get some overzealous T cells in periphery. We need to tell them to calm down, especially if like I'm in my gut or I'm on my skin, where I want to keep some of my microbiome friends happy. All right, let's talk about activation. So those newly matured T cells, all of the last talk was on getting them matured and exiting the thymus. So now they're mature, but they're naive. We haven't found our antigen yet. So they circulate the blood, go to secondary lymphatic organs. So think about what that could be. Give me spleen. Another thing. Well, give me lymph nodes for sure. All right, and a bonus answer, malt. So that's where we're circulating after we have finished the rigorous curriculum that is T cell college. Well, then we're going to find residence in one of these areas, and there's probably a couple other areas that we're not going to go crazy on, but just know that that's the ultimate destination. Go explore the body, find a lymph node, find a spleen, and hang out there. So what they do is then those antigen-presenting cells make their way to the secondary lymphoid structures. So that's your macrophages, your dendritic cells, your B cells, and they present these antigens to the T cells. Do we have that one-to-one match? And let's talk about that. So the T cells require two signals, the two-signal model. So it's a little annoying here, a little complex, not as simple as just the antibody tagging something. And I hate to do it, but we're going to get two more receptors. We're almost, we're getting close to being done with receptors in this unit. Again, so specific recognition. My CD8 T cells binds to my MHC1. So that's my self marker. And then my CD4 likes to bind to MHC2, my other self marker. Just keep it light. We'll go crazy on these next lecture. But just recall that CD4 is going to bind to MHC2 and CD8 to MHC1. A good way to remember that. 8 times 1 equals 1, and 4 times 2 equals 8. Yeah. So just that's some algebra for you to help you remember. Alrighty. Now, apart from that specific recognition, we also have something called co-stimulation. So on B cells, you have, or on antigen-presenting cells, you have an extra receptor that binds to an extra receptor on T cells. So let's go over here. Here's my antigen-presenting cell. So those are going to have MHC, be it class 2 or 1. We'll talk about that later. And that's just saying this is me and this is what I have found in my travels. Then the T cell has its T cell receptor and it says, oh, look at this thing that you found in your travels. This is the exact antigen that I am bred and built to fight. Wonderful. It has a CD4 co-receptor here that also looks at this and helps facilitate the binding. So CD4 matching up with MHC2. Don't worry about CD3. We'll ignore that receptor for now. But also on the T cell, you have CD28. CD28. And on the antigen presenting cell, so this could be on a B cell, on a dendritic cell, we have B7. So this is another linkage that we need. All right. So this is a route that we use to keep us honest, keep good activation honest. If I only get one of these, activation does not occur. So basically what I want you to know is this B7 and CD28 checkpoint here is just one more barrier against hyperreactivity. Even though I found my antigen and my one and only antigen that this T cell is born and bred to fight, it's not good enough. I need to get this cross link here, this double check, to ensure that this is a correct binding. So very picky, very picky. All right. Cytokine reinforcement. So those antigen-presenting cells are going to secrete cytokines, those signaling factors that dictate T cell differentiation and behavior. So depending on how this binding went, this will cause the secretion of different signals that select for different T cell classes, which is pretty cool. All right? So kind of like the idea of class switching in B cells, that changes their antibodies. Well, in this case, depending on the signals present, that will change the type of T cell that I am. Pretty cool. So once activated, clonal expansion, that doesn't change. Obviously, if we are fighting a specific antigen and I have found the T cell that fights that specific antigen, well, I want a crap ton of that T cell. So we multiply him by a lot. And then we differentiate into the effectors, the fighters. So that can be CD8 T cells that go hand-to-hand combat. And it can be the various types of T helper cells, the CD4 cells that are going to go into circulation and manage and field general for me. And then, of course, we also have that subpopulation that goes and hangs out and studies. So puts on the glasses. Let's make him a cell. Why are you oblong? That's fine. Maybe give him a little book. MU. We'll just leave it at MU. Okay, so he's going to go read through his pages and hope and pray that the same antigen comes and tries to fight you again. And hopefully we can dispatch that with a quick response thanks to our memory backlog. So let's talk about the differentiation here. Number one, this one, let's get this out of the way. This is probably the easiest to understand. They are the cell-to-cell combat artists. Their role is to kill infected and or cancerous cells. The mechanism by which they do this, they recognize any intracellular antigens on MHC1. So let me go ahead and yank my MHC1 over here. Come on now. Plop that down. Beautimus. So this is just my minimalistic MHC1 receptor. So this is one of the receptors that we have just presented on most of our cells. When a cell is infected with a virus or a intracellular bacteria, or if it's tumorogenic, your MHC molecules on the cell surface are actually pretty smart. This is the cell's way of just showing the cells around it what's going on inside. So they put little pieces of what's going on inside on their receptor. This is one of these mechanisms that we use to check, is everything inside going okay? So we put those intracellular antigens on top of that MHC receptor, so then any bypassing cells can take a look-see. Say, oh, is that good? Or is something strange going on inside? So that's the whole point. That's the idea behind MHC1. My self-marker presents a little nougat for the rest of my body to assess. And in this case, let's say I am virally infected. That cell is corrupted. The machinery has been taken over by a virus. Give me any virus you could think of. That little chunk there will give me all I need to know that there's an infection going on. So then my CD8 T cell will bring out the daggers. Those daggers, the molecules that I represent are perforins and granzymes. Perforins literally perforate, put holes into a cellular membrane, and granzymes go in and degrade proteins. So the double whammy, poke holes, you're leaking, the cell is completely defenseless, and then we dump acid inside. So very efficient killing process, induces apoptosis. Now if these CD8 cells fail, I can't combat virus infection. Maybe my NK cells will pick up the pace a little bit, but I mean, you don't have anywhere near the efficacy if you do not have your CD8 T cells functioning at full throttle. Also, they're really excellent at combating cancer. Cancer is like you have very, very low amounts as you age all throughout your body, because all cancer is that unregulated cellular division, and you have that going on everywhere. I mean, just think about how many stem cell populations that we've discussed already this semester. That's going to mess up. There's going to be issues that happen sometimes, and you have these rapidly dividing populations in all of your organs. So cancer happens. Just most of the time, it gets regulated. These CD8 T cells say, hey, why are you dividing like that? That's not part of your machinery, and they get rid of you. So CD8 T cells really help keep that in check as well. Again, I love making metaphors and drawings of the different cells, and this is my guy here. So he kind of has the T in his hood there. I think that's kind of dope. And then we have the two daggers representing perforin and granzyme getting in and stabbing and felling the threat that is a virally infected or a tumorogenic cell. All right, next up is the T helper cell, CD4 positive. So the role of these guys is to orchestrate the immune response. They are really just secreting cytokines all over the place. Let me grab my MHC2, bring that on over, and plop it down here. So same kind of deal, just a little bit of a different receptor here. Your MHC1 has a little bit of a different configuration. This more looks like, the MHC2 looks like, some towers. But generally the same thing here. We have the self-marker that presents little nougats that we have found. Just in this case, they are extracellular antigens. So things that have been floating around outside the cell ingests it, breaks it down, and then places it on MHC2. If you're like, what did you even just say? Then we'll talk about that next video. So yeah, more on the extracellular. Let's say. While your CD8 is much more concerned with intracellular infections and antigens. Okay. So we can leave the general CD4 T cell as just this immune orchestrator and the recognizer of extracellular antigens, but that is not the full picture. We have a lot of subclasses of CD4 T cells, so let's talk about them. T helper 1s, Th1 cells, are excellent at boosting macrophages and CD8 cells. So absolutely crucial for intracellular infections. All right, so maybe I'm getting signals that tell my body, that tell my cells that, oh, there's viral infections going on. That's not good. I need to upregulate CD8 T cells. I need to upregulate macrophages because both of these cells secrete antiviral compounds. All right, so that maybe tells me, let's get rid of this extracellular pathway. we're gonna code switch into a Th1 cell to specialize and aid and orchestrate viral removal. So that's the Th1 branch. Th2 is more so parasitic infection. Okay, so if I have signals that tell me I got a dang tapeworm going through my intestines, well, that will tell these generating T helper cells to go down the Th2 branch, and then T helper 2 cells tell B cells, oh man, I have a big old worm going through my organs. I need you to go to IgE. Because IgE, yes, it's very mediated and it mediates a lot of allergic responses, but when it's functioning properly, its goal is to help mitigate parasitic multicellular infections. Another cell that's very helpful for that, eosinophils. Remember from our innate immune system, these things are literally born for multicellular parasitic removal. So that's your Th2 arm, IgE and eosinophils and Th2, all for that parasitic arc. But again, remember, that's one of the ideas behind the hygiene hypothesis. Since worm infection is not nearly as common, there's the idea that this arm of your immune system is bored, and that's where allergies stem from. So maybe we all need to go out and eat a bunch of grub worms. No, I'm just kidding. Please don't do that. Okay, so next up is Th17. These are more so the recruiters of neutrophils and against the defense against extracellular pathogens. So more so like the textbook extracellular bacterial type of defenses. So think back in Unit 1 with E. coli, with pneumonia, with MRSA. That's more so this kind of brand here. And they're really prevalent for barrier fortification. So we see these in most epithelia. All right, so your skin and all the mucosal surfaces that we discussed. All right. Since our country and New Zealand love putting out pharmaceutical commercials, maybe keep an eye out because there's so many skin drugs that are being marketed, and a lot of them, if you keep an ear out now, so Th17 is the one that is the cell that's going to regulate a lot of this. The cytokine, the signal that's involved with this is IL-17. So that's interleukin 17. You don't need to know this. I'm just rambling. But if you keep an ear out now and you see those commercials pop up, you'll see IL-17 inhibitor. All right, so whenever this process is going wrong, that's going to lead to conditions like eczema or maybe psoriasis. And I don't know if I'm just one of one here, but that's all I see are those dermatologic commercials. So be on the lookout for that. And now you know why, because those diseases are a product of this arm of your immune system being unregulated. All right, I already talked about Th2 being unregulated. That's going to start off like allergies responses when we don't need eosinophils, when we don't need IgE. And then we talked about unregulated Th1 already. That's when we have rampant CD8 cells just going through organ tissue, secreting perforin and granzymes. That's compounds used to poke holes and kill cells. That's probably not good to have going around. So again, thinking about not only their function, but whenever the function goes awry, that can give you another idea as to how important it is that it functions correctly and functions well. Lastly, TFH, T follicular helper cells. So these are the germinal center maintenancers. How do you even say that? You know what I'm trying to say. They maintain the germinal center. All right, so they aid in that B cell class switching and affinity maturation. So I think I referenced them just a tad when we were going through this in last unit. The signals provided by these will help determine what antibody profile that I need. Okay, so they can detect if I need to go heavy on the IgG, heavy on the IgE, what have you. So allowing for that increasing in affinity and class switching. So on and so forth. So they help maintain the Thunderdome. All right, if the Thunderdome was all of those cells trying to become the best of the best, these are the referees. These make the rules. All right, almost done. So our last big class we'll talk about is the regulatory T cell. This is technically also a subsection of T helper cells. So these are CD4 in nature. CD4. They have another couple receptors on them, like I think FOXP3 and maybe some others, but don't worry about that. Just know that they are in the CD4 family. Again, for these regulatory T cells, keeping things in check and balance. So immune suppression. All right, these cells, I hope you've grasped this over these units so far. They're so powerful, and they can have a profound effect on the human body. So sometimes we need to tell them to calm the hell down and maintain tolerance against the self and maintain tolerance against our microbial commensals, our friends that help us digest fiber, that help us make vitamins, that help fortify our barriers. So all of that in place is very important. So we want to make sure that we have a population that sometimes calms our immune system down. So secreting suppressive factors, telling the effectors to shut up, and again, tolerance to the self and commensals. Beautiful. We already talked about, hopefully I have a little bit of a memory population formed from this response, so now I don't have to worry about what T cell that I need. I have my whole T cell profile at the ready whenever a secondary infection dares to occur. And then really brief, I don't know much about these because they are unconventional, but you have a different breed of T cells called gamma delta T cells. Those are all throughout your epithelium, like your skin, and they give you that early, like buckshot defense. But I'm less concerned with this for sure. And then you also have NKT cells. This is a hybrid of a natural killer cell and a T cell. Really, really cool. They can really amplify the immune response since they're kind of towing the line, like quite literally, between innate and adaptive. So they can really decide if I'm Th1 or if I'm Th2 really quick due to the cytokine signals that they drop out if they get activated early on in the response. So definitely some cool exceptions to the rule, but don't stress too much on those. I just wanted to mention them for comprehension or to be fully complete. Okay, let's go. To finish things off, to tie it to the macro, the lymph nodes, we'll talk about those a bunch in Unit 4. Primary site of T cell activation. So I don't know, like if you remember last time you were sick, maybe you felt some swollen lymph nodes, maybe beneath your mandible, or maybe in your neck, or maybe in your armpits. Remember, you have lymph nodes in every vertex, every angle of the body. Okay, and that can give you a hint as to where the infection is occurring. That's, again, a lot of Unit 4. We're going to talk about, like, the drainage pathways. So if I have an infection that is bothering, that maybe started in my leg, well, then I'm going to feel my lymph nodes in my hip and my neck, in the closest vertex to my core, because that's where the drainage is happening, and that's where the lymph node was that got an activated T cell to get rid of the infection. So lymph nodes, primary site of that activation. Spleen, think about from Unit 2. Remember, we had the white pulp and the red pulp. Red pulp was literally just blood filtration and storage. So the spleen is great to have some T cells resident there for systemic blood-borne pathogens. I can't think of a better place to check the blood than a literal blood sieve organ. Malt, again, thinking of the benefits that malt gives us, gut and airway defenses, so having some T cells there at the ready, excellent at getting that response handled in the area, hopefully quickly. And like I've referenced, the skin is rich in those unconventional T cells, so that's a great idea to have that rapid responders that are maybe a little bit more specific than the innate immune system, hanging out in the barrier, in like the number one barrier, right? That's the first thing that something's going to have to cross if it doesn't enter your GI tract or your respiratory tree. So it's good to have some sentinels posted up out there as well. So that is my various T cell classes. That's a pretty dense lecture, but I think it covers a lot of good stuff. So if you need any further clarification on any of those classes, please don't hesitate to contact me. And thank you guys very much for your continued attention.